You unlock this door with the key of compassion. Beyond it is another world, a world of science, a world of common sense, a world of sanity. You're moving into a land of both empathy and ethics, of nutritional knowledge and empowerment. You've just crossed over into Dr. Osborne's zone. Hey everybody, welcome to Dr. Osborne's Zone. I've got a very special guest today. Dr. Hazan is with me, and if you have been hiding under a rock, you probably haven't heard about her, but she's been on a lot of media lately for her discoveries in the microbiome. And so I wanted to have her on as a guest because she's finding some very, very amazing things and doing amazing work. So thank you so much, Dr. Hazan, for being with us today.
My pleasure. Thank you for having me.
So let's talk about what you guys are doing in your lab. I mean, if I could just summarize my understanding of it, is you're finding some amazing microbiome changes and risks for COVID and risks for changes in the microbiome post-vaccination. I'd love to hear more in depth about it.
Yeah, I mean, we're at the beginning of all this, right? Obviously, we need like a lot more numbers and everything, but this is exciting because what we discovered through the pandemic was, first of all, we discovered SARS-CoV-2 in the stools, the whole sequence of the gene. And I'm going to call it a sequence because I know there's a lot of controversy on isolation of the virus, et cetera. What I'm talking about is a genetic sequence of the whole virus, right? And so this is, at the level of, mapping the gene of what we were told was the virus and then seeing that gene, different changes in the gene, different mutations throughout the pandemic. So we've been kind of following, this pandemic with isolating, and I don't want to say isolating, but really finding the sequence of the virus in the stools and then looking at the sequence and seeing what the microbiome is doing. So in other words, here we have a sequence of COVID in the stools, and here we have a sequence of a lot of other microbes like, Streptococcus, E. coli, Klebsiella, bifidobacteria, just hundreds and hundreds of microbes that we've identified, right? That is part of that was part of that whole microbiome project where they, had a whole detail of all these microbes that were isolated, right? So from there, we've looked at the sequences of the microbiome and we've looked at the sequences of COVID. And what we noticed was in the severe patients, and so remember, I was doing this before the pandemic, right? I was looking at the microbiome of Parkinson's patients, obese patients, children, old people, young people, everything, a whole different spectrum, vegans, vegetarian, carnivores. So I had a little bit of a good idea of what does the microbiome look like, especially because I do fecal transplant too, right? So, when you do fecal transplant, you take stools from a healthy donor, but you have to kind of know who is a healthy donor, right? You can't just say, okay, well, anybody is a healthy donor, because you and I know that people could appear healthy on the surface, but then, you start doing blood work and tests and you notice they're not so healthy. There's something else, right? So what is a healthy donor? So we started looking at the microbiome of those healthy donors. We started putting it in patients that had a disease, C. diff. And then we noticed a change. We noticed something was happening, right? So we identified C. diff, for example, was really a problem that was just, loss of microbes, loss of diversity. C. diff being a bacteria you get when you take antibiotics, which makes sense, right? Because you're taking antibiotics. Yes, you're killing the strep pneumonia for your pneumonia, but then at the same time, you're killing a whole group of microbes that had a function, right? At least that's what we like to believe, that every species in the microbiome space has a role, right? Much like every species in humanity has a role, right? Not everybody could be, doctors or lawyers or plumbers or contractor, right? Everybody has a role in the planet, and that's what makes the planet beautiful and special. So it's the same thing in the microbiome. It's all these species are all doing something. You think about the ruminococcus, they're doing their thing with vitamin B absorption. You think about the lactobacillus, you think about the bifidobacteria. And so this was my insight onto the whole microbiome space before COVID. So when COVID hit, the first thing that I kind of looked at is I said, well, here we are. The population that is affected are the diabetics, the comorbidities, right? The high risk population. So, and the kids were not really affected. So I said, well, what's the difference between the kids and the high risk population? And really the thing that kind of popped up for me was the bifidobacteria. Why bifidobacteria? Because it's $1 trillion industry of probiotics. So to me, well, if it's $1 trillion industry of probiotics, there's got to be some major data behind it pushing this movement, this business. And so I wanted to look at what's the baseline bifidobacteria in the high-risk patients versus the kids. And in fact, that was the first discovery, was kids have a ton of bifidobacteria and old people have zero. So the aging process is really the loss of bifidobacteria. In my opinion, that was published, that was, written by a lot of other physicians that are in this space. And so that was the first thing. What is the bifidobacteria level in people that have severe COVID versus people that are in the same room, same apartment as people that have severe COVID? What we discovered was the first paper was called the discordant family. So the discordant family was a family of 4 where one kid had COVID and the 3 others didn't. So I analyzed the stools of all those 4, and I realized the kid with COVID had 0 bifidobacteria, and the other 3 had a lot of bifidobacteria. So from there, I started, looking at other families, a baby, a newborn never got COVID. Everyone else in the family had COVID. And it was actually the son of a physician who's an anesthesiologist. So I said, send me all the stools for everyone. And that was the other thing. The baby had a ton of bifidobacteria. The whole family had none. She was a physician. She was, stressed, and her husband was stressed, and there was the whole COVID on top of that, and the whole stress, the news, etc. So anyway, so basically that was the beginning. So when we started looking at high risk versus high exposed but never got COVID, that was the difference we noticed, was the bifidobacteria. And then little by little, as I was looking at the bifidobacteria, and me being always the first guinea pig during the pandemic, is I started, taking some vitamin C. And lo and behold, my bifidobacteria went through the roof, like super high, 4 times the level. So I said, wait a minute, is vitamin C increasing bifidobacteria? And sure enough, I found a paper that showed an in vitro data where they actually took a petri dish of bifidobacteria and they gave it some vitamin C, and the bifidobacteria multiplied. So I said, I wonder if that reproduces itself in other people, right? In humans, right? So I started calling, my naturopaths in Malibu that are, treating with vitamin C COVID, or treating, viruses, or just giving people vitamin C, right? And I said, can I see the microbiome before and after vitamin C, like within 24 hours? And sure enough, we discovered that vitamin C actually increases the bifidobacteria. So it was kind of a discovery. So then when you see that, you go, well, maybe that's how vitamin C is working, right? Maybe viruses penetrate because the bifidobacteria is like 0 in the gut. And therefore, I don't have a good guy defending my immunity, right? So therefore viruses. And that's the whole principle really of gut dysbiosis, right? What is gut dysbiosis? Your bad bugs are high, your good bugs are 0, and viruses penetrate, right? So from there, then of course we, published on ivermectin as a hypothesis, because ivermectin is a product that is from a bacteria that's similar to bifidobacteria. So I started a hypothesis saying, well, maybe ivermectin, which is the same product as bifidobacteria is in the same phylum, maybe ivermectin increases bifidobacteria, because I had observed during the pandemic that every time I gave ivermectin to patients, their oxygen saturation went up, right? And of course, and I know ivermectin is like this big taboo, drug. You can't even talk about it. It's like, zip it. So I basically put the hypothesis out there. I said, look, it's maybe something, maybe nothing, prove me wrong, right? And of course, it was, we had like submitted it like months before it even got into the review process. And then it got published in July 2022. And now 10 months later, just yesterday, I get a tweet that my paper is retracted, because obviously I don't go back to all my papers that I've published to see the, the messages. And nobody, I mean, they kind of, sent me an email, but there was no due process, right? There was no, hey.
There was no, you lacked scientific criteria or data, or
not even like, can you explain to us why you picked these papers, or, what went on in your mind here, or something, along the line, to at least defend, the hypothesis. What was even shocking to me was, why would you even remove a hypothesis? In the title, it says
it's a hypothesis. You're not making a claim.
No, and to me, it's like, science is exactly that. It's about making a hypothesis. Okay, hey, if I'm wrong, I'll be the first one to say I'm wrong. If I'm right, I'll say I'm right. But you know what? I should be allowed as a scientist to make a hypothesis. So this really irked me yesterday, and so, through this whole process of doing research, doing my own research, because clearly, I've been in the field of research, clinical trials. People ask me all the time, why did I step in? I mean, I've done clinical trials for pharma. When it comes to my body, I need to do my own research, because I don't necessarily believe the data. And we've seen the data can be manipulated. We've definitely seen, companies in the past, paying billions of dollars for, corrupting the data. You look at the billions from GSK, you look at the billions paid and fined by Pfizer. These companies don't have a great record of, putting out, good research. And really, it's at the end of the day, it's money talks, right? And so when you look at a company like Pfizer, who's made billions on Paxlovid, are they only focusing on the money, or are they focusing on humanity and health? To me, I focus on my health, and I really, I'm in the field. And so I did my own research for me first. It helped me. I mean, I'm still standing, I'm still talking, and, how many doctors have seen the data, right? I mean, these protocols that I put out on April 2nd or 3rd and 4th, and Peter McCullough put out his own protocols, and doctors were starting to talk, right? Because they started seeing something, right? Pierre Corey, started speaking, and you've seen how passionate he is. And this is a guy that's basically, said to me from the beginning, we are at war, right? I mean, this is not even about a business entity or anything. This is about proving the science, proving the medicine, and this war against ivermectin. I have to ask, this whole retraction, is it because maybe I'm right? Maybe they've seen that and they see that, wait a minute, we could use this for cancer patients. So I'm actually rushing to go to Chicago because I'm presenting data on DDW, Digestive Disease Week, which is in front of all the GI doctors. And the data is actually loss of bifidobacteria in invasive cancer. So, you start thinking maybe we should pay attention more to the microbiome, right? So this bifidobacteria we've noticed is lost in Crohn's naive patients. We've noticed is lost in Lyme disease patients. Now invasive cancer, it's lost. So, how do you rebuild something you've lost? So through this pandemic, and I'm sorry I'm like talking so much and not even letting you speak, but I started looking at the vaccines. And the first thing that really shocked me was the drop in the bifidobacteria by 50% within the first month. That was the first thing. And I said, but this vaccine's supposed to boost your immunity. And if we believe that your bifidobacteria is important in immunity, why is it killing the bifidobacteria? And then I started seeing a couple kids with babies, that were breastfed from moms, and their bifidobacteria was inexistent. Just yesterday, a baby, a year and a half, was breastfed by mom, and it's been a year and a half. The bifidobacteria level is 0.4%. This kid should be having 1 year and a half, should have like 90% bifidobacteria. So the fact that that 1 year old has 0.4%, one has to ask questions, right? So this is my path. This is how I aim to
you follow the evidence as it takes you. You don't cherry pick what you want it to be, and then I think you're right. That's exactly science, right?
And this is not something you can fake, right? This is not something you can manipulate. You cannot send sequences to the sky of microbes, right? I mean, these microbes, you just send it out. Before the vaccine was given in those patients, I had 36 patients, there was a level of bifidobacteria. After the vaccine was given, we had the bifidobacteria drop, not, in the patient. So it's not something you can, then you do, of course, your statistical analysis, your p-values and all that. And you start noticing, wait, the p-value is 0.00082 or 84. This is not something you could just make up, right? I mean, it's not something that's like, okay, maybe I'm wrong. I mean, this is 36 patients with a p-value of 0.0084. You have to pay attention. So
for the audience's sake, a p-value of less than 0.5 is considered to be very, very likely scientifically accurate. So when you get into p-values of 0.00 something, you're onto something.
Yes, yeah. Yes. And that was the thing for me. But not only that, I started seeing also the vaccine injured, right? Remember, I was the first one that was, because I think what happened in this pandemic is we started gaslighting these people, right? Here's a guy that, was flying. I have a venture capitalist. Actually, it's interesting because a lot of venture capitalists came to me, with symptoms. Like, here's a guy that was skiing, parachuting, parasailing, surfing, and then all of a sudden he's bedridden, debilitated, and suicidal, right? What happened there, right? What happened from the moment that before he was fine, right? And then all of a sudden, bam, bedridden, right? And I started looking at that microbiome, and, I'm going to leak it because, unfortunately, we're not in a world where we can wait for the publications anymore, right? Because unfortunately, this is what's happening. If you look at these people, they're lacking bifidobacteria, You look at a kid that was paraplegic and her brother got placebo and she got the drug from Pfizer. Her bifidobacteria is 0. His is high. Here's 2 kids in a clinical trial. One got the drug, one got placebo. One is paraplegic. She has 0 bifidobacteria. So, and it's not only bifidobacteria. Obviously, there are other microbes that go through this process. But I think bifidobacteria, in my opinion, is a little bit more sensitive. It's probably the first one to get this bifidophage, bacteriophage, that I like to call it. In other words, maybe the spike protein goes in and just destroys the microbe. And therefore, that starts a roller coaster, right? So first you lose your BIF, then you lose the next phylums. When I see people that are extremely debilitated and they have one phylum in their guts, I have to pay attention because I have to say, yeah, I mean, unfortunately, that's why this person is like that. And I feel for them, but at the same time, I'm like, I'm not sure what to do with this at this point.
Have you done any fecal transplants in these patients?
You can't do fecal transplant unless you have FDA approval for C. diff. So we wrote the protocol. We're interested in doing it. But unfortunately, that needs funding. That needs to be submitted to the FDA again. When I submitted the 3 protocols for hydroxychloroquine, Z-Pak, and then ivermectin, doxycycline, and then the prophylaxis of hydroxy plus vitamin C, D, and zinc, it took a whole team to write this. And then we had to submit that. And then we had to submit it to the IRB, the review board, that basically makes sure that they're, reviewing the data and they're in between me and the FDA. So, when you do this is a lot of manpower. A lot of people need to write this, and we have to convince the FDA. Here, we can't even convince the FDA. Before we convince the FDA that we can do fecal transplant, we need to publish the data that shows that there's loss of bifidobacteria. But we can't even publish the data if we're still stuck on, hey, a hypothesis is removed, right?
Yeah, they retracted your hypothesis. So, yeah.
So now, like, the journals are going to start questioning the data, etc. So it's kind of like I'm trying to advance the science and I'm being held back with these manipulations of the system to basically stop the progression of science. And the problem with this is what happens is, unfortunately, is science, if you interfere with science, it's going to affect you as the patient, because none of us know it all. Okay. I was fortunate, and I really have to say this is really divine intervention. I was fortunate to look at the microbiome. I was fortunate to have an assay that is valid, verified, reproducible, right? I mean, it could have not been, right? I was fortunate to find COVID in the stools of everybody who had a positive nasal swabs, right? Because what we discovered is you could have COVID in the nasal, but then it can persist in the stools, right? Longer, and you've removed the upper respiratory tract. You think of the kid who never really had symptoms from COVID. Her whole family had COVID. That YouTube video is on still. It's Dr. Hazan and Tourette's. You will see that there's a video that we posted, and just in case it drops, we will be posting it on our website, progenibiome.com, because I made the mistake of talking about the farmer and his wife and how the farmer had a lot of bifidobacteria, and that video disappeared. So anyway, so this kid had, her whole family got COVID. She never had any symptoms, upper respiratory symptoms of COVID. And then in July, about 6 months into the family having had COVID, and I obviously didn't treat her because I was like, she's a kid, she's fine, asymptomatic. 6 months into it, we discovered she had COVID in her stools and she developed Tourette's-like symptoms. So we treated her saying, well, I knew, we treated her. She had 0 bifidobacteria. So obviously she started off either obviously or the virus killed off the bifidobacteria. And I tend to believe it's the spike protein of the virus that does that. But essentially from there, we treated her and then she resolved. Her bifidobacteria went up, COVID disappeared from her stools. So this was a great case to show us that, here's an improvement of a disease that could have been a lifelong disease for this young girl. And here she is improved because we were strategic enough to look at her stools and discover that, yes, she was having COVID in her stools for 6 months. And so this is at the research level, right? So we're doing these tests at the research level. There is no assay. We tried to submit an assay to the FDA for COVID in the stools, and we were denied. And then we tried to submit it. They said, well, there's no purpose in COVID in the stools. So there's no purpose for COVID in the stools. And then we were sent off to the CDC, who basically, was useless. So to me, it was like, wait, there's no purpose in the stools, but yeah, you're looking at the septics. Shouldn't you be looking at the humans and not the septics?
And I've seen a number of studies that show that it actually is not just an upper respiratory infection. It's a gastroenteritis. Yeah.
And I'm going to have to say, just because I'm a GI doctor, and to a GI, everything happens, it's like everything's a nail. I'm going to say it's a GI problem first and then a respiratory problem second. But you heard it here from me. It's going to be, some people are going to go crazy on it. But I really believe it is. You think about these cruise boats where people go in and then they end up having, diarrhea from a virus. And that it went airborne on the boat. It's not because everybody went fecal oral and touched themselves, right? So there's an airborne component. Every time you go to a bathroom, you are exposing yourself to a lot of viruses. Now, whether these viruses are active, whether your immunity is strong enough to fight it, that's the key. That's the question.
So what you said, you had a patient that you treated and had a stellar outcome. What was the treatment protocol that you used to bring up her bifido?
Well, I used to basically, I gave her a little bit, not too much, just a little bit of hydroxy, maybe like 2 pills. People don't realize hydroxychloroquine has a 29-day half-life. So it lasts in your system for a long time. And then the other thing I gave her was vitamin C infusions, and I gave her some ivermectin. Yeah. So
have you found, or have you done this where you've seen vitamin C infusion be more effective for raising bifido, let's say, than?
No, we're still at the beginning of all this. What we're seeing is basically within 24 hours. That's all I can comment on, is vitamin C increases the BIF. And I think the problem is probably what happens is it increases it and then drops, making the body used to drugs instead of fermenting itself. So remember, everything you give your body gets used to it, right? You give laxatives to your body's going to need laxatives. It's no longer going to work to move those bowel movements, right? So everything you give your body, whether it's a drug, you're going to get addicted to that drug. Whether it's sugar, you're going to get addicted to that sugar. Probably you're creating an environment of feeding these microbes, right? And then you adapt them to a certain food, and they either keep on building increase themselves with those foods, or they end up, and hopefully you're increasing the good bugs and not the bad bugs. Because eventually when we die, the process of dying is these bugs take over our bodies and puts us back into the ground. That's the reality of, the microbiome. The microbiome will live on after the humans live on. So we got to be careful. This is a unique opportunity to look at the microbiome. This is a window of opportunity. And I think we got to be careful, and we got to start preserving the good microbiome, because unfortunately, if we don't, it could be the end of humanity. So
no doubt.
We're just a reservoir, right?
Right. I mean, William, you look at pesticides and herbicides and fungicides and chlorine and fluoride in the water, and everything is designed to be antibiotic in nature, and that's what we're eating and drinking.
And we've been programmed to kill, right? So kill, sterilize, alcohol, antibiotic. You have a little cold, antibiotics, even though it's a virus, right? So, we've been programmed to kill. And I think we need to be programmed to add more. It's not about killing, it's about adding the diversity. Stop killing the diversity. So you think about people in the Amazon jungles, you think of people in Central Africa who are killing the zebra, touching the zebra, eating the zebra, blood on their hands, they touch their kids, then they eat the honey, and they don't have diabetes, they don't have cancers that we have here, or arthritis. So, we've gone from that environment to, hey, we got to sterilize everything and live in a bubble. And that's great to live you could live, I always say this to people, you can live in the jungle and survive in the jungle in that environment, and you can live in a bubble, right? But the problem is when you leave the bubble and you go to the jungle, now you're sitting duck because you have no microbes in your gut. So you got to pick where you want to be. You could be sterile, but then you just stick to your sterile environment. You can kill microbes and keep killing microbes in your gut, but then you got to keep doing that, right? Think of Lyme disease, right? Lyme patients. So they catch Lyme because there's probably a gut dysbiosis there. And what we noticed with Lyme patients is they had 0 bifidobacteria, right? So what happens is we keep giving them antibiotics, and they feel great while they're on antibiotics, right? Because you're sterilizing the gut. So there's no bacteria or viruses that's going crazy. And the majority of them are getting like doxycycline, for example, which has a pretty broad coverage on a lot of things. And then you stop the antibiotics, they're back to where they were. So, so that's, and that's the same thing with Crohn's disease. That's the same thing with a lot of diseases. We got to start going into the world, looking at the microbiome and looking at what are these drugs doing, what are these pesticides doing, and from the general view that I see, where we look at just autism, for example, you will see that autistic children, we had, in 1980, in my era, 1 in 2,000 kid was autistic. Now it's 1 in 44, 1 in 36, but they're predicting that in 2030, it's going to be 1 in 16. So what are we doing there, right? What is happening? Obviously, we're not doing something right if we're heading, and this is the problem. You ignore these kids. Who's going to take care of the old when those kids need to be taken care of themselves, right? So we've got to stop the destruction. And it's not only autism. I wish I could say, well, yeah, it's just autism, but it's Alzheimer's, it's Parkinson's, it's cancer. I mean, all these diseases are just keep on climbing. Maybe we need to rethink and say, maybe we were wrong. Maybe, yes, you can kill the microbe for the disease, but then understand that you're killing other microbes that causes other diseases. And so we need to start looking at the microbiome of all these diseases, which is what I'm doing, to understand what is each disease pattern of microbes, and not doing it as a commercial lab, but as a research, because it's still research, because there's still billions of bugs that we have no idea what their names are, what they're doing. Think about, a tomato plant has a bunch of viruses in it. People don't even know that. A pepper has a lot of viruses in it. So if you're immunosuppressed, I had a kid that I found was immunosuppressed, and, he was exposed to these pepper plants, right? And his gut had a lot of this virus that was in the pepper plant, and he was having fevers like once a month. You eradicate that offending, you build up the immunity, and then the kid doesn't have fevers anymore. So there's a whole world of viruses and bacteria that we have no idea what they're doing. So my job as a clinician is to look at the clinical data and to look at the bugs and say, maybe this bug is the culprit of this, right? Kind of like what I did with that little girl with Tourette's was, let me look. I went on a gut instinct, and really it's all gut instinct, which I call divine intervention. Let me look at that girl. She was exposed to her parents with COVID. Maybe COVID was in her gut for 6 months doing ravages, right? And so that's really what it's all about. And I think if we stop the science, we stop the research, we stop getting into this world, it's gonna affect all of us, because at some point we're gonna be the patients with Alzheimer's, Parkinson's, cancer. And then we're gonna say coulda, shoulda, woulda. So I don't want to say coulda, shoulda, woulda. That's why I stepped into it at a time where I could have easily retired. I'm the wife of a cardiologist. I'm a physician. I've done fine. I've had businesses. Could have retired, done fine, right? This was my time to kind of say, okay, guys, it's time to see the data on the microbiome.
So where can people find your research? I know you publish, but is there a layman's place where our audience can go and read more about what you're doing?
Yeah, so let's talk shit. That's the first step. I saw no grain, no pain, but mine was like, let's talk shit, because I published it at the beginning of the pandemic, and to me, I felt like this was a shit show, pardon my pun. And I said, you know what? Let me talk. I know they're going to say I'm full of it, so I might as well beat them to the punchline and say, yes, let's talk shit. So, and to me, I wrote it because it's very educational. A lot of people have liked it because it kind of gives them hints. We try to stay very, simplistic for the layman. I think, I'm the type of person that gets stuck in the verbiage of the medical, right? And I'm sure you too, you like to have simple things. I like people to explain things to me in a simplistic manner, because then you get caught. And to the person that's non-scientific, non-medical, when I say anaerobic bacteria, they're like, what's going on? But if I say, look, bacteria that doesn't breathe oxygen that's stuck in your colon, that's a different field. So we kept it very simplistic. So that's one thing. We're going to write the Let's Talk Shit 2.0, which is basically the whole all the discoveries through the pandemic. And then the other thing is the publications on progenibiome.com. We have a whole dedicated area for publications we were linking the publications with the journal, but now after yesterday and the retraction, we decided we're just going to put the PDF of the paper so that, you know, people can have access, even if the journal decides to retract it. You know, I'm working with Dr. Peter McCullough to start our own, you know, ethical journal with open access, you know, for the public, for the doctors that want to see all the data, right? We shouldn't have to cherry pick these journals, right? It shouldn't be the highest bidder pays for the journal in New England Journal of Medicine, right? So think about all the papers that are published in New England Journal of Medicine, Lancet. There's always a lot of money behind those papers. It shouldn't be that way, right? A person that's a scientist in a little lab somewhere makes a discovery. We should have access to those discoveries. And then we, as the critical physicians, you know, decide, hey, you know what? This is valid or this is not valid, right? But I think that's where we need to kind of, you know, go in an open access for the public to do their own research. You know, I'm an open book because to me, you know, I like transparency. I like to know what's in my yogurt. I like to know what's in the injection from the vaccines, right? We still don't know that, right? So that kind of, you know, that was a red flag for me because you're giving a vaccine to the rest of the world, to the whole entire world, and we're not allowed to investigate as an independent lab what's in it. What if there was a contamination, right? Then who's looking at that? We're not allowed to test the vaccine vials. That's not okay. That was not okay. So I believe in transparency. You know, we want to continue doing research. We want to continue to educate the public, and you know, that's me with this microbiome venture. It is research, so it's extremely expensive. We did a case of autism where we did fecal transplant, and remember, fecal transplant is not approved except for C. diff. And we did it with the FDA basically approving us, but that case ended up costing over $300,000 just of regulatory and paperwork. So could you imagine if you had to do one after the other? But we had great success with that case. Now we need to do more. We need to raise funds. So I'm constantly juggling. How do I raise funds? How do I bring in funds? How do I, you know, juggle continuing to do this the right way, the ethical way, you know, because I think at the end of the day, you know, nobody can buy us to change the research. If you change the research because you were paid, you just altered the fate of humanity, in my opinion, right? So, I mean, I'll tell you, at the beginning of the pandemic, I was offered $10 million for Progenibiome. Then it went up to 40 million, and then it went up to 185 million. So, you know, but I didn't sell because I'm like, Progenibiome is Sabine Hazan, and it's Sabine Hazan guiding this boat to see the data first, right? And then to have my colleagues, more importantly, come on board to say, hey, you know what, you're right. That is legit. Let's reproduce it. Let's see. That paper with the loss of bifidobacteria in high-risk patients was actually reproduced in France, Italy, and China. All research needs to be valid, verified, and reproducible. If you don't have reproducibility by an independent person that doesn't know Sabine Hazan, right, then it's not valid. It may not be valid. It may just be a fluke that you discovered that. But the fact that in different countries, China, Italy, France, they found loss of bifidobacteria in severe COVID patients tells you that it's valid. And again, this is not research you could just fabricate, right? This is research that is genetic sequencing that goes to the cloud and gives you a that you put in through your bioinformatic pipeline and basically tells you, hey, this is what we found, right? So, you know, this is where we're at. So supporting us would be great. Progenibiome has a link for support. That would be great. But essentially, we're going to try to make it as possible as as accessible as possible. Would love to do with every paper that comes out. If I get to publish again, who knows? I'm gonna have to put my husband's name on the papers, or have him. I have like 9 doctors in the family. Surely one of them will be publishing, and I know like over 300 doctors. So I'll give the research to be published by someone else. But the data needs to be published and needs to be out there. So I'm happy to come back and talk and present in layman terms. So you could be the dedicated podcast for layman.
We'll do it. I know you have a hard stop here, but I'd love to have you back on and dive into poop a lot more. I've got a ton of questions for you and a ton of, just a ton of insight I'd like to ask your opinion about. But thank you really for being with us. And I know you're super busy and doing great work. So keep up the great work. I'm going to put links below for people to come visit your site and check out the work that you're doing. And let's get you back on again whenever you can spare a moment.
Thank you. And thank you so much for having me and being patient with coordinating all this.
Of course. I understand.
And you too, keep doing great work. So we need good doctors. We need good healers. So,
amen.
All right. Thank you so much.
Have a wonderful day.
Bye-bye.
Bye-bye. Thanks for tuning in to the Dr. Osborne Zone. Don't forget to share, like, and subscribe for more content like this. And make sure you come back next Tuesday at 6 p.m. Central Standard Time and Thursday at noon 30 for more episodes.